All right, good to see you all. Let's go ahead and get started, shall we? So today, what I want to do and what we're going to do is we are going to finish up our talk about inheritance. So I have a talk about inheritance. We're going to start with X-linked inheritance. And then a lot of you asked for a supplemental lecture on blood typing. So I made that, and we're going to go through that. And then I have some details about the exam. So we'll go over some details about the exam, which opens up today after lecture at 10:15. All right, let's get started. Okay, so just share my screen. All right. Oh, sorry. My cat is making noises. Okay, so let's start here. And then I will just want to see. Hold on just a second. I got to get my cat out of here. Just a moment. Sorry about that. She can be very loud if she has any doors shut. Okay, so let's talk. And just a reminder, if you have any questions as we're going through, please feel free to ask them in the chat box. I'm happy to answer them. All right, so we've been talking about inheritance. So just a reminder that we talked about primarily, we've been talking about single gene traits or traits that are inherited in a Mendelian fashion, meaning that there are basically two alleles for every trait, dominant and recessive. And then the combination of how those are inherited determine the trait that it's expressed. We talked about that. We talked about variations on Mendelian inheritance. So, what, for example, incomplete dominance, where you have instead of having the heterozygote express the dominant phenotype, the heterozygote expresses sort of like an intermediate phenotype, so shown by the flowers. Like if you're dominant and recessive are red and white respectively, the heterozygote would be kind of the in-between of those, or pink. We talked about codominance, which is shown in the chickens, where basically there's no recessive necessarily, and both alleles are dominant. So therefore in the het, both of those would be expressed. So that's the black chicken and the white chicken, and then the heterozygote has both black and white feathers. Multiple allelism, we're going to expand on that today when we talk about blood typing. Multiple allelism is when there are more than two alleles for a certain gene. So we talked about this in the context of blood type, which we're going to expand upon here in a minute. We also can talk about polygenic traits, where we have multiple genes influence a phenotype, which kind of explains anything that has sort of like a gradient, like height, eye color, as you can see in this image, autism spectrum, etc. We talked about pleiotropy, where you can have like one gene influence more than one thing. So our example we talked about this was sickle cell anemia, where the gene actually codes for sickle cell anemia or not, but it has influence not only on developing sickle cell anemia, but also susceptibility to malaria. And then we just mentioned briefly gene linkage, where multiple genes are linked and travel together during crossing over of meiosis. So what that would mean is that they're inherited together. They're kind of linked. So we talked about this in the context of red hair and freckles. And then lastly, we talked about environmental effects. We talked about how temperature can affect expression of certain genes. So for example, in Himalayan rabbits and Siamese cats, how colder areas turn on genes for darker fur. And that's why you see darker fur around the nose or ear regions. We will expand on environmental effects on genetic expression patterns in the next lecture when we talk about epigenetics and some other really cool stuff that can happen because of the environment. So we talked about all those variations. I want to expand on a couple more variations today. Because we are on this slide, what I'm going to do before we move on to like X-linked inheritance, because this is the slide where we talked about codominance and multiple allelism, what I want to do is kind of jump on over to the other lecture that I made, which is posted on Canvas and has to do with blood typing. So I wanted to talk about blood typing because it is a lot of you are interested in it, which I love, and that also it gives a really good example, again, of multiple allelism and codominance. Plus, it's just a really important thing to know about in general, especially right now where we are in need of blood donations. So let's talk about it. So first off, I kind of always like to talk about why blood is important, even though we haven't quite got there. That's what we're going to be covering in Unit 3 after we talk about genetics, or finish up our talk about genetics. What we will talk about after we get out of sort of the genetic. As for notes regarding this, there is no questions on the exam about blood typing. This is just a good example. Thank you for that question. Let me take that back. So there's no, I typed that in. So what I mean by that is there's no direct questions about like this slide or blood typing in general, but we will have questions about inheritance of blood types. So that was already a part of the cellular reproduction and inheritance lecture. So this is just an expansion on that, but there won't be any direct questions from this that weren't covered by the other lecture. So thank you for asking that question. I should have specified that. All right. So why are red blood cells important? Why does blood typing matter? One of the big things about that blood does is, and there's a lot of reasons red blood cells are important, but one of the major reasons red blood cells are important are because of the fact that they carry oxygen and carbon dioxide through the body. So again, we haven't talked about oxygen and carbon dioxide and its role in the body yet. We will get there, but I'm sure all of you are aware that oxygen is extremely important in the body. The reason it's important in the body is because it is what is used to create ATP. So we'll talk about ATP, we'll talk about cellular energy, but it's just worth noting that basically like the body cannot run without ATP, and we can't make ATP without oxygen. So oxygen is basically, along with sugars like glucose, like we know about, oxygen is extremely vital for energy production, which fuels every single thing in the body. As well as that, a byproduct of those energetic processes is carbon dioxide. So whereas we have to get oxygen to cells so they can do stuff, those cells kind of like, as a byproduct, create carbon dioxide, and then we have to get that carbon dioxide away. And so red blood cells do both of those things. So red blood cells, you can see on here, it says hemoglobin. Red blood cells all over have a protein called hemoglobin, and hemoglobin is what kind of holds on to oxygen or carbon dioxide. So when blood goes to tissues, when it gets there, you can see the purple arrow. What it does is it donates oxygen to tissues and kind of allows those tissues to have that oxygen generate energy. But then once they generate that energy and have the byproduct of carbon dioxide, you can see with the black arrows that that's loaded on the red blood cells. And that's carried back to the lungs, and then you breathe it out. So that's why they are so important. So again, your body cannot run without oxygen. Your body cannot run if it cannot get rid of carbon dioxide. Carbon dioxide is acidic or contributes to acidity. So a buildup of carbon dioxide would lead to an acidic environment. And as we already talked about, we have to maintain homeostasis. So we have to get rid of that carbon dioxide. Take oxygen in, get rid of carbon dioxide. And red blood cells are the cells that do that. So they are an absolutely essential cell in the body. We, as you know, have blood types. So red blood cells are not universal, meaning that my blood cells may not be compatible with your body. Your red blood cells may be not compatible with my body, and that all comes down to our genetics. So a little bit about blood typing. And the reason we're talking about this here is because blood types are a wonderful example of two concepts that we talked about, and that is multiple allelism, where there are more than one allele for a trait. So for example, for blood types, you have 3 alleles. Technically, you have A, B, and O. And we'll talk about what that means in a minute. This means that there can actually be a lot of traits, a lot of phenotypes for blood type. You can have type A blood, type B blood, type AB blood, or you can type O. So we'll talk about that. Blood typing, if you are type AB, is also a really good example of codominance. And that's because when we look at the blood types, type A, which as you can see on this slide, A is a dominant allele. So type A blood could have the genotype AO, which is heterozygous. A would be expressed because O is a recessive allele. Type A could also be homozygous dominant for A. Type B blood could be BO, a heterozygous dominant B, or it could be homozygous dominant B. Type AB would be an example of codominance, like I mentioned, so you would have both dominant alleles, A and B. And type O blood would mean that both recessive alleles were present, so that would be homozygous recessive. So what does all this mean? So you can sort of see off to the right, hopefully everybody can see this, what we're talking about when we're talking about A, B, and O. And what we're talking about is what are called antigens on the surface of these cells. So antigens are just, as I'll talk about in a minute, they're little protein kind of markers on the cell surface. And as we talked about, there are for blood cells 3 different versions of these antigens. So you can have type O, which you can see here in this picture is just kind of this red, yellow, blue pattern. It's just one little protein. Type A has the type O antigen, but then has a little red box off to the side you can see here. So technically type A would just be that little, I guess that's a hexagon coming off the yellow fragment right there. So type A, this red box or red hexagon, sorry, would be the A antigen. This is the O antigen, A antigen. So in this case, that would be type A, AO technically. And then you would see type B, which has O, and then you see it has a yellow hexagon. So that yellow hexagon is technically the B antigen. So type O just has the O antigen. Type A type AO, sorry, would have the OA antigen. Type B, BO antigen. And if you see type AB has both of those dominant traits present, you can see that it has two antigens on the cell surface, A and B. So why does this matter? Let's talk about it. So I do want to mention, and we'll get into this later, that as somebody asked last week, we also have another component aside from just A and B and O. We have the plus and minus. And that's coming from what's called rhesus factor, or Rh, and sometimes this is called the D antigen. So we'll talk about that in a minute. So I made this slide to try to make things a little bit more clearer with what we're talking about when we're talking about these antigens. So just a reminder, antigen is just a protein on the cell surface. So if you look on the left for an example, let's say you have type AO blood. So what I have here on this A cell is I have an antigen right here, and then I have just the A antigen. So AO means that the only dominant allele is expressed, so we have A. That would be type A blood. And we have that protein sticking off, is that protein is the A antigen. Hold on just a second, sorry. Allergies. Horrible allergies. I do have. So that would be an antigen, a protein on the cell surface. So those are really important. So I hope you remember when we talked about cell structure, and we talked about how cells have certain patterns of molecules on their surface. So when we talked about that in lecture, we talked about the glycocalyx. And the glycocalyx on cells is like a sugar pattern on the outside of a cell. But importantly, that sugar pattern is pretty unique. So the glycocalyx is another example of something on the surface of cells that kind of tells us what that cell is, where it came from, and who it belongs to. So antigens are the same. Technically, the glycocalyx could probably be considered sort of a cell surface antigen, cell surface marker. So this is just another way, again, antigens are just another way for us to determine, you know, again, like what is this cell, where is it from, and who does it belong to, especially blood type antigens. So how do we recognize whether it belongs or not? We recognize where anything in our body, basically our immune system is a surveillance system that its sole purpose is to try to recognize whether something belongs or not. And it does this in kind of a complex way, although it's a very efficient way. So if you look here, so let's say that below the line on this slide, this is the body of somebody who has type A blood. So like we know, if a person has type A blood, let's see their genotype is AO, they have the type A antigen expressed on their cell surface. So that means that in their body, type A antigen belongs. That is correct, right? The body recognizes that as good. And basically, the way the body recognizes something that is good is by ignoring it completely. That is very important. So last week, somebody asked about autoimmunity. Autoimmunity is when your body fights itself. Autoimmunity would be a breakdown in immunity. So like I said, in order for your body to recognize that something belongs, it ignores it. With autoimmunity, what happens is your body stops ignoring stuff that belongs. What's what we call rejection. So we'll talk about that. So if you see that line, we see that type A blood person would have type A antigens, and that's basically just, it belongs. So it's not recognized by the body, it's just like good. This person who would have type A blood, because they have type A blood, which is again ignored because it belongs, if they were to receive, let's say like a blood transfusion, and that blood was type B blood. So you can see that off to the right on the slide. If you had the wrong type of blood donated, that would mean you would have what's called a non-self antigen present. So if you look on the left, remember that this type A blood, which is type A person's blood is right, we call their antigen that belongs to them, so again, type A person with the type A antigen, a self antigen. It belongs. If that person received a donation of the wrong type of blood, that antigen would be called a non-self antigen. I think sometimes this is also referred to as like a foreign antigen. So basically it's an antigen that does not belong in the body. So in this case, the immune system does recognize this as bad. It doesn't belong. And so when the immune system recognizes something, what it does is it creates immune molecules, proteins, and these are called antibodies. So you can see in this picture, I have it in green. What happens is your body starts to produce antibodies to the blood type that doesn't belong. And you can see that antibodies recognize antigens. Antibodies sort of look like a Y structure, so we always draw it like that. So your body will always have antibodies created to the blood types that are the opposite of yours. So for example, if you were this person, if you had type A blood in your body, you already have type B antibodies, right? So they exist. Your body has already created antibodies that recognize something that is bad. So what happens when an antibody recognizes an antigen? We'll talk about it. But first, I want to talk about the different blood types that exist and what that would mean for the people with those blood types. So this is just looking at all of those different blood types. So again, blood type is determined by what antigens or the proteins on the surface of the cell. If you have type A, you would have A antigens on the cell surface. If you have type B, you have B antigens on the cell surface. If you have type AB, which remember, that's a codominant phenotype. So that means that you would have both A and B antigens on the cell surface. And then you have type O. So notice, look at type O, all right? So that figure earlier was a little bit confusing. How do the antigen proteins protect the red blood cells? Let me talk about that in a minute. That is an excellent question. Right. So if you look at type O, you'll notice that there are no antigens on the cell surface, right? So O is a recessive phenotype. And even though that one slide kind of showed it had like a little protein, realistically, because that protein is basically what the A and B antigens attach to, this figure I like better because it simplifies it a lot more. But because that's what the A and B antigens attach to, basically O would be present on every single red blood cell regardless, right? So just to remind you, if you look back at this, right, type O is here on type O blood. Type O is on A as well. Type O is on B as well. So really, when we're looking at blood types, what you're looking at is that kind of little supplement, A antigen or B antigen. O is sort of, you know, everywhere, so no one recognizes it. So sometimes you would say that an easier way to look at that is just to see like type O is sort of like a naked cell. It doesn't have A or B expressed. Okay, so those are our antigens. Type A has A antigens, B has B antigens, type AB has both A and B, and type O has nothing. All right, so remember that whatever blood type a person is, that person will have antibodies or immune molecules that recognize the blood type that doesn't belong. So therefore, type A person with A antigens would have antibodies against type B blood. A person with type B blood, where B antigens belong, would have antibodies against type A blood because A doesn't belong. If a person has type AB blood, they have both A and B antigens present, and therefore they would not have antibodies to anything, right? So they actually don't have antibodies. This would be somebody that's called a universal recipient. We'll talk about that in a minute. And then you have type O blood, which doesn't have either A or B antigens. This would mean that this person has antibodies to both A and B, because type A blood would be recognized as non-self, type B blood would be recognized as non-self, type AB blood would be recognized as definitely non-self. So this person would have antibodies to everything. So this person would be restricted to only being able to receive a donation of type O blood, because everything else they would recognize as non-self. However, the benefit here is that because they don't have any antigens on their cell surface, they could donate to any of the other blood types. Because although all the other blood types do have antibodies, there's no antigen on the cell surface of type O blood. So type O is what's called a universal donor. So I have that summarized on the next slide as well. So here is a very big slide, but it gives you all of this information about blood donation. So again, you can see type A blood has type A antigens, produces antibodies against the B antigen. And because of that, remember, it has antibodies. So a person with type A blood would have antibodies against any blood cell that has the B antigen present. All right. So what this means is because they have antibodies that attack B, they can receive blood from a type A person, right? Because they are type A. But they can also receive blood from a type O person because type O doesn't have B antigen either, right? So they can receive from both of these blood types. Type A blood can donate to another type A individual because that's compatible. They could also donate to a person with type AB blood, right? They could donate type A blood to a person with type AB blood because a person with type AB blood would recognize both A and B as safe. So if they get type A blood, they're not going to say this is wrong, right? Because they don't have antibodies to type A. So you can go through that. Type B, same thing. Type B has B antigens, produces antibodies against A. So what that means is that a type B person could receive from type B, but because they have antibodies against A, they could also receive type O because there are no A antibodies. They can donate to somebody with type B because obviously that's compatible, but they can also donate to somebody who is AB because a type AB person would have no antibodies, so they wouldn't have any antibodies to fight type B. Type AB blood is a little more complex. It has both A and B antigens. Because of that, it doesn't have any antibodies. And because they don't have any antibodies, basically that means that they can receive blood from any of the blood types. So they're called a universal recipient. So they don't have antibodies, so they have nothing that will fight a different blood type. However, they are restricted on who they can donate to because if somebody has type A blood, they'll have B antibodies, which will recognize type AB. If somebody has type B blood, they'll have anti-A antibodies, which will recognize type A blood. So, and type O has antibodies against A and B, so they wouldn't be able to receive type AB blood either. So type AB is nice because they can receive any type of blood, but it's a little bit more restricted with who they can donate to. And then type O is called a universal donor. And they're called a universal donor. This is one of the blood types that's usually most in need. The reason it's most in need is because type O, again, does not have any antigens on the cell surface. Because it doesn't have antigens on the cell surface, they can basically donate to any other blood type, right? Because no matter what blood type a person has, if they have type A, they're producing anti-B antibodies. Type O doesn't have any antigen that can be recognized, so it can be donated to anyone. However, they are restricted with who they can receive blood from. And again, that's because due to the fact that they have no antigens on the cell surface, they recognize both A and B as foreign antigens. So what that means is they cannot receive type A blood, they cannot receive type B blood, they cannot receive type AB blood, because all of those would be recognized as foreign. So the only blood that type O can receive is type O, right? So as I mentioned, and I haven't forgot your question, I promise I will get to it in a minute. So as I mentioned, sorry, this is very loud. As I mentioned, and as it was brought up last week, A and B is not the only type of antigen that is present on red blood cell surface. We also, have to worry about another antigen on the cell surface, which is that RH, or rhesus factor, sometimes called D. And that is a little bit more simplistic, and it's basically just, is it there or not? And it is a dominant phenotype, so it's basically like homozygous dominant or heterozygous will have Rh positive. So you'll see Rh positive just means that there is an antigen there, whereas homozygous recessive would mean that it just isn't there. So this would add another layer of complexity. We'll talk about it here in a minute. So the question was, how do the antigen proteins protect the red blood cell? And the answer to that is a little bit complex. Antigen proteins don't necessarily help protect the red blood cell, but what they do is let me just write this down because it's a little bit more, basically like what antigens do as a category is they help kind of identify a cell. So basically, what is the cell? It's a red blood cell, right? Where does this cell belong? In the plasma, you know. And then whose cell is this? You know, that's your type. So it kind of identifies the cell. So antigens are not necessarily like protective necessarily so much as they are for identification. What the protective component of this is all about the immune system. So technically the immune system is protective and that it just recognizes self or stuff that belongs versus non-self, which does not belong. So we're talking about blood typing, but obviously this is far more complex. So for example, like, although this is a little bit complex with blood, because you would hope that, you know, human blood would be compatible with other human blood, but evolutionarily we are related to a lot more organisms, right? So not only does blood typing and blood cell antigens, all this stuff help distinguish A versus B versus whatever, it also helps distinguish species. So there's species differences, which even though it's very, you know, like who cares if I was to get pig blood versus, you know, human blood, whatever, you would hope that that would be okay. the evolutionary benefit of this, if you will, is that it would basically help you recognize pathogens or not. So a pathogen is a disease-causing organism. So for example, you mount an immune response to anything that doesn't belong. And that immune response is actually what causes you to not feel well. So right now, obviously, we're in the context of a pandemic, so it's pretty easy to say like a virus would be a pathogenic organism, a disease-causing organism. It does not belong. What happens if a person gets that virus is the immune system recognizes viral antigens. So those are antigens on the actual virus that we recognize do not belong. And what happens is we mount an immune response to that. The immune response is powerful, hopefully it works, and would therefore eliminate the organism. But when you feel sick, when you don't feel good, that is a result of your body's immune system mounting that response. So it's the same thing here, although I wish I could say that your body is complex enough to say, well, a different blood type isn't that big of a deal, whereas a virus is a big deal. It's just at this point so hyper specific due to the variability of pathogens that exist that your body just says anything that doesn't belong, we're getting rid of it. I hope that answers your question. So it's a little bit complex. Could you explain how plasma from a previously infected person could be used to help some newly infected people? I could. Let me get to that in a minute. That's an excellent question, very applicable right now as well. Let me get to that in a minute. Okay, so now we know, again, kind of how this all works, that if you have your blood type, that is referring to the antigen on the cell surface, and that you make antibodies to anything that doesn't belong, right? So for example, if you have type A blood, you're gonna have antibodies to type B blood because type B does not belong. So, and we kind of went through how each blood type. So, two questions. One, how do we determine a blood type, right? So if you have somebody's blood, how do you know that it's type A or type B or whatever? And this will help us answer your question, which is, could you explain how plasma from somebody previously infected with like a virus or something could be used to help somebody else? So this will answer that question as well. Okay, so how do we determine blood type? We do what is called an agglutination test. So first off, let me reiterate this picture right here, where antibodies recognize their matching antigens. So this is taken from the slide that says a type A person produces B antibodies because B blood is non-self for them, so they create antibodies to something that does not belong. And those antibodies, though, recognize that thing that does not belong. So what this means is that antibodies recognize their compatible antigens. So if you have B antibodies, those recognize B blood, all right? So we can use this technology to determine, or use this knowledge to determine the blood type of a person. And we use it for like a million other things, antibodies and antigens, as we will talk about in a minute. It's probably antibodies and antibodies in general are probably like one of the most important biologics and biologic kind of ideas for science. This is used in all fields of science, I would say. So again, antibodies recognize antigens. Anti-B antibodies would recognize B antigens. The person that created those anti-B antibodies would have type A blood, okay? So what happens when an antibody recognizes an antigen, you can kind of see in this picture here. So this picture is a little bit hard to like wrap your head around, but I'm going to orient you to it. Remember that antibodies are this Y shape. So the reason they have this Y shape is because each of those little arms of the Y can actually grab an antigen. So you see here, this is a red blood cell with the B antigen. This is a red blood cell with the B antigen. This would be a person who, because we see it has the A antigen and the RH antigen and the B antigen, this would be an AB positive blood type, right? So we have these B antibodies, and you'll notice that each arm grabs a B antigen. What happens is red blood cells are normally, and if you look in this picture up here, this would be a blood spot from somebody whose blood does not have antibodies that recognize the antigens. We'll talk about that in a minute. But red blood cells are normally sort of like evenly spread throughout the plasma. But what will happen is if you start to get antibodies that are grabbing red blood cells, those start to clump. And that clumping is called agglutination. One of my favorite words, agglutination. So you can see what happens if you look in these two test tubes. If you see this A test tube and this B test tube, right? So what you would do is you would have a person with, you would put like the blood, the unknown blood in here, basically. And the A and B would be indicating what kind of antibodies you're adding. So let's say that in this tube, we put the blood in here and we added anti-A antibodies. And you see that the blood is still consistently red. It's sort of, those red blood cells are still suspended. This would mean that their anti-A antibodies didn't recognize anything. Okay. And then you see in this test tube, this is a bad example, but I didn't make this figure, so ignore it. But here we have the blood and we add anti-B antibodies. And since those antibodies recognize the red blood cells, they started clumping them up, right? So you see the red blood cells have started clumping. And what happens is it kind of like clumps all those red cells out of the plasma. So what happens when an antibody binds an antigen is it binds all of these, anything with the antigen together and makes a big clump of it. All right, so we'll get to that again, Anil, your question in a minute. So what we can determine is if we had a test tube of blood, right, and we added anti-A antibody and it clumped, that A antigens were present on that red blood cell, right? So this is a little bit opposite because we're not talking about production of antibodies, right? We're talking about what antigens are present on the cell surface. And we're using antibodies that we have in a lab to determine that. So this is what we would see on a slide. So for example, what you're looking at here is recipient's blood, so the person, and donor's blood. So the person that would be creating the antibodies would be the recipient, right? So let's say that a person has type A blood, the recipient, type A serum. Remember, if they have type A blood, they would create B antibodies. So if we had a donor give type A, we have B antibodies. So those B antibodies in a type A individual would have nothing to recognize. However, if this person with anti-B antibodies was given type B blood, they would recognize type B blood, those type B antigens, and you can see what happens is we have that agglutination, which is that clumping. Same with type AB blood, because of the fact that those B antigens are present, type A people make anti-B antibodies, you get agglutination or clumping. Okay, does that make sense? All right. Yes, Eric, the test is called an Eldon card. You can get them online for $10. That's true. Yes, you can do that. Also, if you donate blood, just a little pitch. If you donate blood, they will tell you for free. All right. So that's what happens when you have an antibody recognize an antigen. So Anil, your question was, how can you explain how plasma from a previously infected person can be used to help newly infected people? So for that, I'm gonna like draw something. All right, so let's say, actually, here we go. Let's say that this is a virus. All right, so let's say in the context of something. All right, so viruses, like I mentioned, have antigens just like everything else. These are going to be, because this is a pathogenic organism, a disease-causing organism, it doesn't belong in anyone's body. So everybody, let's see if I can do this. So everybody, because this is a foreign antigen, is going to create antibodies against it. All right, so there is our antivirus antibody. So basically anybody that gets infected with a virus will mount an immune response to that virus. Part of that immune response is creating antibodies. These are very specific. These are antivirus antibodies. All right, so what we can do, because that would be present, right? Sorry. Let's say this is our person 1. Who got infected. And let's say that this is our person 2 who is not infected. Okay, because they have not been infected, they don't have any antibodies, all right? So first off, let's talk about what happens in person 1 under ideal circumstances. In person 1, under ideal circumstances, what would happen is if this antibody bound the virus, the same thing would happen that happens to the blood, is it would clump up, okay? So what happens obviously this is Gen Bio, so we haven't talked about this, and unfortunately we probably won't, although I'm working on a lecture for you specifically about the virus, so I'm going to work on that. But what happens is if your body starts creating antibodies, the virus actually has to use antigens to attach to your cells to get inside of them. So what happens when you start creating antibodies to the virus is you're binding all of those antigens, and without those antigens, the virus can't enter the body's cells. So since antibodies are actually present just in the plasma, what we can do, let's say that person 1 got infected with the virus, they created a bunch of antibodies to the virus, and that led to the virus being eliminated. The virus is gone. They're no longer infected, all right? So let's say previously infected now. Even though the virus is gone, they still have all of these antibodies. So what we can do is draw this person's blood and get their serum. Their serum would contain these antibodies. And what we could do is donate it to this person. And now this person would have these antibodies, these antibodies generated in somebody else, but still recognize a foreign antigen. And then if they were to get the virus, automatically they would have these antibodies to neutralize it. So what happened in person 1 is that they had to generate the antibodies. This takes time. This can take a week or two, all right, to generate those antibodies. If we draw the antibodies and if we donate to somebody that has the virus, they don't have to wait 1 or 2 weeks to generate those antibodies. They're already there. They're already available to fight that virus. Right. So that's the benefit. The only issue with this is what is called passive transfer or passive immunization. And these antibodies are typically only good for about 30 days. So this is not the technology that we use for vaccines, where we try to get a person to like generate long-lasting immunity. This would be, as opposed to like a preventative, this would be more of a therapy. Right, so the person already has the virus or the person's at risk of getting the virus, so let's give them these antibodies to either neutralize the virus or hopefully in the short term prevent them from getting the virus, neutralizing it. So I hope that answers your question. That's an excellent question. So basically, like we talk about agglutination, this is how antibodies recognize antigens. We can determine blood type with this. It agglutinates and we can just see the blood type, determine the blood type visually, but in the body, agglutination is a very, very, very important part of immunity, because the exact same thing happens when it sees a foreign antigen, is it clumps it all up. And if it clumps it all up, it can't function. Awesome question. All right, so another question, are you saying that vaccines have other technologies so they can last longer? Yes. Absolutely, yes. So you have, let's see, another side. So like vaccines have 2 forms basically, or I guess I should say immunizations have 2 forms. You can have vaccines, which would be active, and then you would have basically passive immunization. So vaccines, what they do is they give you some sort of inactivated or dead or something version of the pathogen. This leads to an immune response, and the person develops immunity and immune memory to the pathogen. So for example, when you are a baby and you get a vaccine to, like, let's say whooping cough, you get, I believe the whooping cough is, I can't remember, but it's either dead or inactivated. So you're given this vaccine. The vaccine is actually like a dead or inactivated fragment of the organism that causes whooping cough. So it's Bordetella pertussis. It's like a little dead or inactivated fragment. It can't get you sick, but what it does have is it does give you all of the antigens present on that organism. And if you have those antigens, you're developing antibodies. So the benefit to vaccines is they are given in advance of the disease, and thus prevents the disease because the person develops memory, so in their body. Passive immunization, on the other hand, is basically the transfer of antibodies generated in a lab. So this would protect via inactivation of the pathogen, like we just talked about, like agglutination, and there's some other things that antibodies could do, but it is basically only active for the half-life of the antibody, which is about 30 days. And this is given as a therapy, because it doesn't establish immunity or what's called immune memory. So because all you're giving them is antibodies, those antibodies can degrade, so it's not long lasting. In order for a person's body to fight a disease and remember how it fought a disease, it has to establish memory. And the only way to do that is for a person to like actively see the antigens, which has to happen through a vaccine or through the actual infection. So the question is that why people usually only get chickenpox once? Yes, that's a little bit, chickenpox is a little complex. Yes, you can get the chickenpox vaccine or you can get chickenpox. And once you get it, you'll mount an immune response to it, but your body will remember that virus, varicella, and it basically will, next time you encounter it, it will remember how it fought it so that you won't develop the symptoms again, right? You already saw it. So shingles, that's what I was going to say. Chickenpox is a little bit more complex because it can actually, once it goes in the body, whether you get the vaccine or the actual infection, once it gets into the body, it can actually, like, your body can fight it, but then it can like hide in your nerve cells. And then something later on, like, that would, your immune system basically keeps it at bay. It's like, okay, like you can stay in the nerve cells, but you better like hide out there or else I'll get you. Maybe something like dampens your immune system. Let's say like a common one is stress. So a lot of people get shingles during like finals or like right before a big game. I remember like our most valuable player, I played college volleyball, so our most valuable player got shingles like right before a really important game. And it was because she was so stressed about the game. But if you have something that dampens your immune response, your immune system is sort of like not able to keep it at bay. So because chickenpox can hide out in your nerve cells, what happens is if your immune system kind of like takes a break, it can come back. So you still have the immunity. It's just that that virus in particular is just kind of like sneaky. It's a really good question. So I guess like kind of what we're sort of like talking about with this, and kind of probably what Anil was getting at is like right now, one of the things that's being explored as a hopeful kind of like thing we can do in the immediate future to hopefully kind of like combat coronavirus is hopefully take serum from people that had coronavirus and transfer that to people who need it, and give them the antibodies that would neutralize coronavirus. This, again, is like a therapy, a short-term fix. It would be amazing. So I do know that they're working really, 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 really hard on trying to do that. It takes a lot of time, just like any other vaccine or drug, lots of safety. As you can imagine, a person's blood contains lots of antibodies, and then generating these antibodies in a sufficient quantity takes a lot of time too. But that's what we're hoping to do. Good questions. I love it. Microbiology. All right. I am working on a lecture for you guys for the virus, so please know that I am working on that. Okay. Excellent questions. So moving on, just for the sake of time, I did post this on Canvas, so you can see here. This is looking at like all of the blood types that exist. So A, B plus O, looking at the antigens present, the antibodies present, how it would look if doing the blood typing, who they can receive to, who they can donate to, just if you're interested. I also put this in here, which if you're interested, this game actually won a Nobel Prize, and it goes into the technology or the science behind blood typing, and it can allow you to kind of practice that as well. I encourage you, if you have the time, to do it because it's fun. I did it last night and I was like, wow, this is too much fun. I need to do other things. All right. So we're going to move on. Are there any questions about blood typing? All right, if there are, actually, let's like hold them. You can type them in, but I'll probably wait to answer them because I just want to get through a couple more slides for you. Okay, so we talked about that. Let's move in to a little bit more about inheritance. So I want to talk about some kind of specifics of inheritance because of the fact that we've been pretty much talking about inheritance where there are alleles present on two homologous chromosomes, right? So you have two copies of each chromosome in diploid cells. These are homologous, meaning they're the same genes, maybe different alleles. Remember that in human cells, we have 23 pairs of chromosomes. 22 of these are called autosomes, meaning they are non-sex coding chromosomes. But the 23rd pair are sex chromosomes. So females have 2 X chromosomes, males have 1 X chromosome and 1 Y chromosome. So that would mean that a female would have 23 pairs of homologous chromosomes because the 23rd pair, the sex chromosomes, 2X. But males only have 22 pairs of homologous chromosomes because the 23rd pair of the sex chromosomes are X and Y, so they're heterologous. So that's important, by the way. Remember that. Okay, so I mentioned earlier that there's something called a dose response with different alleles. This kind of goes into that. So with the majority of your chromosomes, you kind of have like the combination of 2 chromosomes contributing to the traits. But when it comes to the X chromosomes, because of the fact that females have 2 X chromosomes and males have 1 X and 1 Y, what needs to actually be contributed to a human is actually just traits from one of the X's. So basically what happens is that in female mammals, one of those X chromosomes is inactivated. So we talked about this a while back when we talked about generation of bar bodies, how females will kind of like have those cells, like, we'll talk about what happens to them, but they basically condense into unusable information. We'll talk about how that happens. But what this does is it means that every human only has genes or traits coming from one of their X chromosomes. So this obviously makes sense in males because they only have 1 X chromosome, but in females, you have 2 X chromosomes, but your traits are only coming from 1 because the other is inactivated, generates a bar body, just like we talked about with those egg cells that are generated through meiosis. And in fact, this is such an important component of inheritance that if a person were to have 2 active X chromosomes, that's lethal. So X inactivation is absolutely required. So this is kind of just an example of that happening where you can observe it. Calico cats, you can see that this happens, that their coat color is actually determined by, or I guess female calico cats, which calico cats are only female, because coat color is determined by the X chromosome. So you can see that early on, these cats have homologous X chromosomes, and they will have maybe 2 different alleles, so one coding for orange fur, one coding for black fur. And what happens is throughout development, X inactivation occurs, but it will just be random which of those X chromosomes is inactivated. You can see here a patch of these, the black fur carrying X chromosome was inactivated, and here the orange fur patch was inactivated, and that is what results in this sort of like patchwork calico color, is that X inactivation. And again, that's another reason that, so males don't undergo X inactivation because they only have 1 X chromosome. So that's why there are no male calico cats. All right. We already talked about this, but I just wanted to mention again that that I feel like, did we talk about sex-linked traits? Did we skip this slide? Oh my gosh, I'm so sorry. I want to come back to this slide because it's like we did not talk about sex-linked traits. Okay, so we talked about X inactivation. So this slide is strictly talking about genes that are coded for on the X or Y chromosomes. So sex chromosomes, again, the X and Y chromosome, it's important to note that the X chromosome, and you can see in this slide, the X chromosome is much larger than the Y chromosome. That's because it contains way more genes, and a lot of them are not involved in sex determination. So like we just talked about that in cats, the X chromosome contributes to fur color. So a lot of these are not involved in sex determination. And that makes sense because all organisms, male and female, have X chromosomes. So we can have a lot of genes that are involved in other things aside from sex. And that's why they can also carry more genes than the Y chromosome. Y chromosomes are smaller. They contain fewer genes. Almost all of these contribute to male development, such as the hormone testosterone, which is one of the kind of big pushes for male development. And specifically, the Y chromosome will specify that the organism will be male. So what this means is that females are female because of the absence of the Y chromosome. And this is obviously not present in females. When we talk about sex-linked traits, so for example, like color blindness, sex-linked traits are frequently on the X chromosome, and they affect males more than females. And I have this why, but we're running out of time, so I'm just gonna tell you why. The reason this is because whereas females have 2 X chromosomes, if they have a disease on one of those X chromosomes, that X chromosome can be inactivated, and so they would have the non-diseased X chromosome lead to expression of traits. That doesn't always happen, right? There are colorblind females. However, it's more present in males because if they inherit an X chromosome with, for example, color blindness, they only have that one X chromosome. They don't have another. So if they have it, they have the disease. So with sex-linked traits, in females, their expression is determined based on X inactivation. But in males, it's just if they get it, they have it because they only have that one X chromosome. Okay. So again, just a reminder that not all genes are inherited on chromosomes. You can also have extranuclear inheritance. We talked about the mitochondria. Plant cells, this also includes plastids like chloroplasts. Here's some examples of human mitochondrial diseases. Don't worry too much about it. I did want to point out that last year in Nature, they found out that mitochondrial DNA can also be inherited from fathers. I need to read this. This paper was a little bit, I'm not really sure, but that's sort of preliminary data. And then there is your study guide. So we have 5 minutes left, and what I wanted to do is tell you a little bit about the exam. So I made this to just kind of go over some details about it. So for the Unit 2 exam, it opens today at 10:15, so very soon, so you can start working on it immediately if you want to. And then it closes on a week from today at 11:59 p.m. So you have an entire week to do it, but I'll talk about what that entails. It is open book and open note, but I would like to dissuade you from using Google, and also it is meant to be, I want you as an individual to come out of this feeling more confident in your knowledge and capability surrounding inheritance. So I really hope that you will use this as an individual learning experience, not a group learning experience. So getting into the two parts of these, if you go to our Canvas page, there's two places that you can access this. I think I left this up. There's two places you can access this. First, if you go to our home page and you scroll down, you'll see Unit 2 exam. You'll see a link to the Unit 2 module. You'll also see the quiz, which is part 1, and the problem set, which is part 2. You need to do both of those. I don't care what order you do them, but I have a suggested order. All right, but that's where you can access it. You can also access it if you just click on Unit 2 and exam module, and it'll take you to the Unit 2 module. The discussion is active, and I will be active on that all week as well for questions, etc. I encourage you to use that. So you'll notice it's not 100 points, it's only 95 points. I do want to do like a most missed question thing where you can get 5 extra points. I'll talk about that later. But the test is divided into 2 parts. So the first part is a quiz. I say first part, it doesn't mean you have to do it first. Actually, I would probably recommend saving the quiz for last, because I do think that the problem set will help you learn and hone your techniques a lot better before doing the actual quiz. So let's talk about the problem set first in that case. So the problem set is 65 points. It's a series, it's just like a part A, part B, part C worksheet. What you need to do is download the worksheet, do all the problems. The problems will require you to do some Punnett squares, look at some genotype and phenotype predictions, and then it will also teach you a technique called the chi-squared goodness of fit test. We didn't talk about this in class because what I want you to do is learn it through the activity. When you complete the worksheet, I want you to use colored text or highlighting just so that your answers are very apparent. And then what you'll do is you will upload it to this assignment. So you'll click on this. It kind of has the details. You can download the assignment here. And then once you do that, it'll have a button on your end that says submit up here. All right. So that's the problem set. Again, I would recommend doing that. The reason I give you a week to do this is because I want you to give you a week realistically for that problem set. For the quiz, I do think that problem set will help you on the quiz, although it is important to note that on the quiz, you will not have to do, I'm going to type this in, no chi-square goodness of fit questions will be on the quiz. Those are only associated with the problem set. So you'll know what I'm talking about. When you do that problem set, I will upload this Exam2 details to Canvas as well. The only stuff that's on the quiz is stuff that's coming directly from like the lecture. Yeah, from the lecture. So you'll have to do some Punnett squares. You'll have to do some genotype, phenotype stuff, but you won't have to do a chi-square goodness of fit test. That's just for the problem set. The quiz is 30 points, 1 point per question. It's multiple choice. It is timed. So you have 60 minutes from the start, so you can't stop and come back. It gives you 1 question at a time. You can't go back. It's random question order as well as kind of like randomized answers. And you do have to use Respondus Lockdown Browser. So that means, again, you can totally use your book and you can totally use your notes for that. But I really would dissuade you from using Google or internet searches for that because of 2 things. One, it's going to be a waste of time, right? You only have 2 minutes for a question. That may seem like a lot, but some of these you're going to have to do like Punnett squares for. And then 2, you need to study for this as if you were studying for like an in-class test, because you need to have the knowledge on hand and ready for these questions. It's not excessively hard, right? It's not beyond what you're definitely capable of. I have 100% confidence in you. It's just, I would use your textbook and your notes as sort of like an emergency rather than being completely reliant on them. All right. So that's a little bit about that. So would I recommend doing the problem set prior to the quiz? Absolutely. Absolutely, yes. I could not spell absolutely. But yes, I would definitely recommend doing the problem set and saving the quiz for later. I think the problem set will help you a lot to study for that quiz. And then take your time studying for the quiz. Again, you don't have to take it until, like, you could wait until you have 60 minutes. You could wait until, I wouldn't wait beyond 9 p.m. next Tuesday, but, you know, take your time, study, be prepared, be confident in monohybrid, dihybrid, mitosis, meiosis, cellular reproduction and inheritance is what we covered. So, and we covered it a lot. With this, you'll see this caveat. So obviously, like, there are sites. I've never offered this test before, so I don't think anything like Study Blue or Course Hero is really going to help that much. But just know that using those websites is considered a copyright violation and plagiarism. So don't use those websites. The university does check that stuff to see if it's up there. Again, I do want this to be an individual activity, and so instances of obvious, noticeable sharing or copyright, or instances where that's believed to occur, it's a 0. If I see anything, or if they report to me that anything's on Study Blue, Course Hero, all of those things, that is a copyright violation. So they notify me of that stuff. So don't make me worry about that. So between now and when this is due, we have, I have office hours today. We have lecture and office hours Thursday. Next Tuesday, we have lecture and office hours. So lots of lecture, lots of office hours to ask me questions. But also, if those times don't work, absolutely send me an email and let me know if you want to meet. I obviously can't give you the answers, but if you need help, I am like, I want to help you. The purpose of this is not to punish you. That's why tests are such a punishment. But it is not meant to be a punishment. It is meant for you to demonstrate your knowledge. And if you're struggling, for me, the important thing is that you learn this. So reach out, right? If you need help, reach out. That is very important. So I got a private question, but I'm going to answer it for everybody because I think it's a good question. The question is, if you have your book and notes online, will you be able to answer that? So for the quiz, because it uses the Respondus lockdown browser, you wouldn't be able to access it like wherever you're taking the quiz. So what you might have to do is you would have to have like the quiz open either on your phone or something, and then your notes or ebook open on another thing. If that creates like an undue issue, reach out, right? If you don't have the capacity to do that, let me know. I would recommend using like your PowerPoint slides or written notes and stuff like that if possible. But again, if you have some sort of extenuating circumstance, listen, I want you to succeed, and I'm not, I do need to make sure that you understand it, but I am not here to make your life hell. I'm here to make sure you learn biology. So let me know that. Can you access a PDF on your computers with your notes on the lockdown? I think that's the issue. Yes, I believe so. Let me check with that. I think with Respondus, what it prevents you from doing is opening another browser tab, like Googling anything. But let me check if you can open like PDFs or other files. That's a good question. I'll figure that out. I'll find out. All right. So class is technically over, but, right, if you have some more questions, ask them, right? If you need to go, go. But I can hang out for a couple more minutes if you need me to, if you have questions. If you have questions, just go ahead and type them in. Or if you, you can also make it to where you type in a question privately, it's whatever. But if you can't think of a question right now, I will also have my Zoom office hours from 11:30 to 12:30, and you can certainly email me at any time. I am here for you. You got this. I know you can do it. Right. Oh, thank you. All right. If I don't have any questions, what I'm going to do. Oh, I had a question. Ooh. The question is, just to verify, during the quiz, you can have open notes. So during the quiz, you can have open notes. Again, it uses Respondus Lockdown browser. So I don't know whether you can access like the ebook in a different browser. That might be a question. But you can have like physical notes, you can use your book. I need to see if you can access things that are downloaded onto your computer. That I'm not really sure. But during the quiz, what you shouldn't be doing is like Googling or trying to find it in that way. So again, when I say it's open book and open note, what I would recommend doing is going through your PowerPoints and going through your stuff and maybe making like a study guide, you know, make a physical study guide based on the terms and the concepts and write it out on paper so you can have it physically in front of you. The reason that is important, right? Study as if it was not open book and open note. The reason that is important is because it's 2 minutes per question. And I went through and I took this test, right? Like one of the questions is a dihybrid cross. And if you can do that in 2 minutes, like good for you, but I can't do it in 2 minutes. So at least there's only one dihybrid cross, but that takes longer than 2 minutes. So you need to make sure that your notes and your textbook are just like a just in case thing, and that you feel confident that you can answer these questions without them. So I would recommend, that's actually my best recommendation, is instead of like relying on searching the book or searching whatever, I would recommend doing this, filling out the study guide on like a physical piece of paper and having that be a reference. The terms, huge. Being able to do a Punnett square, huge. Stuff like that is how I would recommend doing that. I hope that answers your question. I guess, yeah, okay, good. All right. Any other questions? All right. You know where to reach me. I am 100% here. I want to hear from you. I'm sharing my screen. I got it really good. I'll send that out. I got a good, whoa, that's not what I wanted to do. I want to share everything with you guys, but probably not my email right now because it's terrifying. So, okay, so if nobody has any questions, I'm going to log off. I will be back from 11:30 to 12:30. You can also reach me via email. Just to show you, you know, my emails do pop up on my phone, right? So I get them right away. And yes, the third slide on this PowerPoint. I will also upload this to Canvas so you can see it. But yeah. Right. So if there's no other questions, I'm going to stop screen sharing and stop recording, and I will talk to you soon. All right. I will see you guys soon. Reach out if you need. Don't worry. And I hope everything's going well. Oops. If I can do